Well, good morning, everyone. Welcome to our fourth webinar as part of the CPATH Rare Disease Program. Today, we're going to have Jamshid Arjomand from the FSHD Society to have a discussion about the gaps in clinical trial readiness for FSHD. First, a few cleaning items. Next slide, please. So this webinar is recorded. It will be accessible online once it's ended. We will have a Q&A session at the end of the webinar, and we will have other panelists join us, including Jamshid colleague June and Lisa Fink from Nord. If you have questions, please place them in the Q&A in the chat box, and we will address these with our panelists at the end of the webinar. Next slide, please. So today, Jamshid Arjomand is joining us. I'm very glad to have him present today and discuss the critical needs for FSHD. Jamshid is a PhD with background in neuroscience. He has more than 50 years of experience in pharma and biotech. Prior to joining the FSHD Society, he worked for a biotech called Genia Biocells. He was managing the scientific direction and efforts to expand the company's pipeline. That pipeline included a drug that got orphan drug approval by FDA for FSHD. After that, he worked for, before that, he worked for the Huntington Foundation, CHDI, where he was managing a portfolio of academic, clinical, and industry-driven projects. He comes to the FSHD Society and is going to take the lead from now on. And Jamshid, if I missed anything relevant, please tell us. Thank you.
Thank you very much, Alex. Really appreciate being on and being asked to present on behalf of the FSHD Society about some of our work. I'd like to introduce also my colleague June, who joined us. June is senior director of patient engagement and research. So she's at the interface between research and patient engagement, and she'll join us in the Q&A panel. So I believe I'm going to share my screen now, and hopefully that will work. Hopefully you can see. You can see my screen. Is that everything okay? Fantastic. So I can advance now. I figured I would start off by giving an overview of the FSHD Society and who we are as a rare disease patient advocacy group and what we're trying to accomplish. I'll give a brief overview of the indication of facioscapulohumeral muscular dystrophy. It is a rare disease. I figured that maybe some of the attendees may not be familiar with this type of muscular dystrophy, even though it is one of the most common muscular dystrophies, genetic muscular dystrophies out there. And then the steps that the society took in trying to identify gaps in clinical trial readiness. And I'll try to give an overview of how the field has really advanced over the last couple of years. And this is hopefully what is happening for all rare diseases, but the steps that we took. And the obvious need for data integration and a broader overview from and the types of data that might be available, and the integration approaches that we've taken. So with that, who are we? The FSHD Society is the world's largest research-focused patient advocacy. We're a nonprofit, and we focus exclusively on facioscapulohumeral muscular dystrophy, or FSHD. And our mission is really to find treatments and a cure for this disease while empowering our families. So we really have a patient advocacy part where we're trying to educate and do a lot for our patient community. And we're also investing a lot in clinical trial readiness. And this is from basic research all the way to patient engagement, education, so they can participate in various studies. And we we've started this industry partnership with pharmaceuticals in our space, and that will be the bulk of the presentation today. So on the advocacy side, what we've done, and the society's been around for, I believe 30 years, and really predates me. I joined only in 2019, but there's always been this patient advocacy and patient engagement component to it. And several years ago, before my arrival, we started having patient chapters, and this is throughout the country where there's a mass of patients. We've gotten them organized so they can provide educational material, what's happening in the field, updates, also provide support for the patient community. And we've now expanded these chapter programs. We have a lot of educational ones. So we have what are called FSHD 360 conferences. These are whole day conferences that take place in conjunction with a clinical trial research site. So the local patient community can come and meet the expert caretakers and the KOLs that provide care for FSHD patients, and they can learn a little bit about what the research and what clinical trials are taking place. We also quite often have visitors and participants from different pharma companies that come and give updates on their FSHD programs. We have monthly webinars called FSHD University, where we provide a host of different topics, and June can talk about that during the Q&A session. We provide standards, we'll have an expert physical therapist or a nutritionist and different people that will come and give insights on how people can better manage their disease. We have a quarterly publication, and obviously we have a lot of blogs and podcasts and a lot of activities on social media. And lastly, I wanted to touch about the World FSHD Alliance. Even though we are situated in the US, we actually collaborate with a lot of other organizations, and we are trying to provide resources as well as share experiences with all the different international FSHD patient organizations throughout the world. So we have very close alliances with all of the different countries that are highlighted on the map on the right. And it's really, I think it takes a global effort to really have an impact. And this is how, through this World FSHD Alliance, we are helping to organize all the different groups so we can be stronger with one unified purpose and try to help the community. So what is FSHD? As the name implies, it's Facioscapulohumeral. This is how the disease was first described, and it's for the majority of the patients, the manifestation of the disease really starts in the upper body with weakness in the face. So people cannot smile, maybe they cannot close their eyelids all the way down. It affects the shoulder girdle, and the arms. But although this is how the disease was first described and it's what's named after, modern imaging tools have shown us that actually the entire skeletal muscle system in the body is affected and can be involved. And so there are a lot of core muscles that are affected, a lot of the leg muscles are affected, and quite often some of the leg muscles are affected before the upper body ones are affected. The disease itself is very asymmetric, although this image doesn't show it. It's not unusual for patients to have a weaker side, it's also progressive, and it will worsen over the lifetime of an individual. So more and more muscles will get involved, but the rate of it is it's varies, highly variable. So it's very heterogeneous manifestation, and it's asymmetric in its presentation. So what are some commonalities, and what is known about the disease? Well, prevalence or incidence of the disease is not exactly known. There are some publications out there that say it's 1 in 8,000. So it's either can be anywhere between the third most common or the second most common genetically caused muscular dystrophy. It's probably highly underdiagnosed because there are people who are manifest the disease very lightly, and they never get diagnosed. They manage, they live their entire life with these deficits without really ever knowing that they have the disease. The genetics of it are quite complicated. I'll go over that a little bit later on, but it is autosomal dominant, and it is believed that maybe 25% of the diseases are spontaneous mutations, although that is debatable, because when we speak with neurologists and they diagnose a patient and then they meet the parents of the patient or other family members, they notice signs in the patient. So we really don't know if it's probably both underdiagnosed, and there are some cases of spontaneous cases. The manifestation, if you look at the population-wide, currently it's believed that about 25% of patients will lose mobility at some point during their lifetime. The disease is not considered to shorten lifespan. However, it's highly debilitating, and probably more than 10% of patients at some point will require respiratory aid, either non-invasive respiratory aid. And there are also cases of early onset that affects, that manifests much earlier in children, as seen in this image. But these cases tend to be more severe and they progress faster. So, what are some other signs beside muscular dystrophy? So you have the obvious facial weakness, scapular winging. I mentioned the core being implicated. So Beaver sign, this is when the belly button starts pointing up because of contraction of the upper muscles of the abdomen. There's a lot of foot drop, a loss of ambulation. You can have weakness in the back causing lordosis and curved spine. And then other typical symptoms that are seen quite often is pain. Most patients report bouts or continued pain and burning sensation, chronic fatigue. As I mentioned, there is respiratory weakness for quite a few patients. There's also seems to be a symptomatic hearing loss, that more patients have hearing loss than the general population. And in rare disease, in the rare cases, and usually mostly affecting younger affected patients, children, there's Coates disease, which is a rare retinal vascular disease that can be actually fixed. So this is very important for early manifest patients to be looked at by ophthalmologists. So what causes FSHD? The genetics are a little bit complicated. I'll try to simplify this as quickly as possible. Let's sum up that the disease is caused by the misexpression of DUX4 and its toxic gain of function. So DUX4 is a developmental gene. I'll touch that on a little bit later on, but and the DUX4 is expressed at a very particular time during embryonic development, and after that, the area at the end of chromosome 4 becomes highly methylated, and the chromatin contracts and compacts, and so you don't get any DUX4 expression, for the rest of the lifetime. So what happens in FSHD type 1, there's two types of FSHD. You basically get a contraction of these repeats that are 3,300 bases long. And in a healthy individual, there are about 11 to 100 of these D4Z4 repeats. But in affected individuals, this number drops to about below 10. And in addition, and in the last D4Z4, what you end up having, there's a gene that gets encoded called DUX4, and the most distal D4Z4 reads through this last, in the last repeat, DUX4 actually gets read through, and there is a sequence that can either stabilize the polyadenylation site. And if this haplotype is present, then stable transcripts of DUX4 are made. These are translated then into DUX4 protein that causes the downstream effect, the toxic effect to skeletal muscle. So the genetic diagnostics is quite complicated. These repeats are present throughout the chromosome. They're also present on chromosome 10, and there's variants of these repeats on other chromosomes. So it becomes quite challenging to diagnose, to do a genetic diagnosis for FSHD type 1. FSHD type 2, which is a more rare form, about 5% are affected, are mutations in genes that are important for DNA methylation, such as SMCHD1 and DNA methyltransferase 3B. And mutations in these, basically you get hypomethylation of these repeats. And if you have the 4qa haplotype that stabilizes DUX4, DUX4 could get expressed and be translated into DUX4 protein, which is then toxic to skeletal muscle. Now, the expression of DUX4 is very low. It's been estimated to be anywhere between 1 in 1,000 to 1 in 5,000 myonuclei might express DUX4, but that's sufficient to be toxic to the muscle fiber. So as I mentioned, DUX4 is a developmental transcription factor. It is expressed when the embryo goes from a 4-cell to an 8-cell embryo, and this triggers a set of developmental pathways, and it activates hundreds of genes. And there's quite a few reviews on this topic, but one of the more recent one kind of looks at the different gene pathways that can be activated. And you can see as an early developmental gene, there's a lot of pathways that are that get activated, and consequently there are a lot of downstream causes that might cause toxicity to muscle. And as you know, in muscle fibers, in myotubes, you have the myocytes fuse to form these myofibers that are multinucleated, and just expression of DUX4 in one of these will then, as a transcription factor, it can activate a series of genes, and we really don't know what causes the damage to the muscle and ultimate loss of muscle fiber. But it could be anything from oxidative stress, activation of the immune system, or even just apoptotic cascade within the muscle fiber. So that's kind of an interesting kind of segue then to what are the possible modes for intervention. So obviously there are a lot of activities happening early in trying to regulate and trying to prevent DUX4 from being expressed. So there are groups that are looking at trying to increase the methylation, so DUX4 will be repressed. There are signaling pathways upstream of DUX4. As you know, transcription is highly regulated, and there are drugs that are being tested right now that prevent DUX4 transcription from happening. Obviously, there's always the promise of gene editing, where we might be able to eliminate DUX4 in muscle cells and prevent DUX4 from being expressed. There's also a great deal of activity in trying to destabilize DUX4 transcripts. So the mRNA, and there are a lot of ASO approaches, antisense approaches, morpholinos, microRNAs that might regulate the stability. And so this is a very promising avenue for trying to reduce DUX4. And of course, downstream, there are some very innovative methods being developed by trying to interfere with DUX4's activity as a transcription factor. So DUX4 has a bunch of cofactors that it binds to. There's still a lot of basic research being done trying to identify those and seeing if the cofactors could be inhibited. And there's also quite a bit of effort, mostly from the academic sector, looking at downstream signaling and how DUX4 signaling could be inhibited to try to mitigate the disease progression. So with that, I would like to kind of give a historical overview of what is happening in the field. When I joined in 2019, there seemed to be maybe about 4 or 5 companies that I knew of that had active programs to try to find a therapeutic for FSHD. Today, we can count over probably 20 biotech and pharma companies that have active programs. And this is absolutely incredible. I think that a lot of the groups, a lot of pharma companies, biotech groups are seeing FSHD as a tractable disease, and they are developing very innovative ways to try to to solve this devastating indication. And there are this is a figure is taken from our website and just highlights a couple of different companies and where they are in their drug development program. The drugs that are being developed are a combination of small molecules, either novel molecular entities or even repurposed molecules. There's also a lot of biologics, viral deliveries, antisense deliveries, and a lot of them, most of them are actually targeting DUX4 in one way or another, but a few approaches have been developed at trying to boost muscle regeneration. And this is not really addressing the DUX4 cause of the disease, but it is a way of increasing strength and possibly improving the quality of life for the patients. So around the time that I joined the society, we had June and my colleagues had organized this meeting we called the industry collaborative meeting, and we invited all the pharma companies that we knew were active in the space, as well as those that were interested. We also invited a lot of the KOLs, clinicians, and scientists in the field, as well as the regulatory agencies. And there were really three points for this meeting. One of them was to introduce FSHD as an indication to the regulatory agencies and to get their take on maybe what needs to happen to help the companies that are actively working in the space. We also wanted to promote FSHD as a tractable indication to the companies that might be on the fence. And most importantly, we wanted to find gaps in clinical trial readiness, so we could, as a society, try to organize kind of a consortium approach to trying to solve these, because a lot of these gaps we felt were going to affect every drug development program, and being able to solve them as a group would help the entire field. And it would also de-risk it for the companies that would normally have to solve every one of them on their own. So what were these gaps? Just broad terms, there was a need for tools, especially biomarkers. As I mentioned, DUX4 is only expressed in very few cells in the muscle, so even muscle biopsies are not always indicative of whether DUX4 is being expressed in that muscle biopsy or not. So looking for a blood-based or a circulating biomarker is very important. Imaging was emerging as a really important tool that kind of gave us a more holistic view of what muscles might be affected and how heterogeneous the disease might be, and that still is in development. Obviously, for any sort of a successful clinical trial, there needs to be a clinical network and infrastructure to run these trials. And the society had been involved in setting up the clinical trial research network with 4 sites early on. And this network had now grown to about 8 sites in the US. And there was a push to kind of expand that beyond the 8 sites in the US. And there were also additional European sites being added. And now I think there are close to 20 sites that are part of this network, and it's still growing. So this is a very important critical aspect, is to have that infrastructure, and as a network, they can actually test and start doing some important research and testing out outcome measures that could be used in clinical trials, and I'll touch on that a little bit later on. And of course, another great important aspect is to engage with the patient community. And this is actually, we heard this from the FDA, that it's really important for them to hear directly from the patients. And they had created this program called a patient-focused drug development meeting. And they encouraged us to organize one of these, which we did in 2020. We had about 600 participants in this one-day meeting with the FDA. And we have a report that's now been sent to them. But another important aspect is to understand the natural history of the disease. As I mentioned, the disease is heterogeneous, it is asymmetric, and trying to get a better understanding of the natural history and getting enough numbers of patients involved in these studies is really paramount to understanding the disease. And as I mentioned, also the genetics are quite complicating. So to try to facilitate testing and simplify it, and providing patients with better access to testing was very important. So for the rest of the presentation, I'll focus on understanding the natural history of the disease and the efforts that we've taken. So there are several different sources of data, as you can imagine. For almost every rare disease, there's usually registries get established by clinicians that really focus on the disease. So they are typically housed at a university, and the registries try to keep track of disease progression in a cohort of patients, whether it's regional or national. So in the US, we're lucky that there's been a registry for patients to participate in. It's been going for probably 20 years or more, and has and it's an annual patient-reported outcome. So they receive a survey that they fill out, and then this is entered into a database. So in 2016, I believe, my colleague June organized this meeting to basically get all of the different registries involved, so they would collect a core set of data. And this was published in this paper, and all of the participating registries are collecting similar data across these core data sets. And right now, Treat NMD, I think, is working on the update these core sets with possibly more refined questions. One of the things that June has done is, I think a year or two ago, she reached out to the existing registries internationally to kind of get an understanding of where they are and what maybe some of their bottlenecks are. And as you know, no surprise, there's limited funding to keep the registries, and the clinicians don't necessarily have the time to input all the data into a standardized format. And you know, as we take a look at these type of registries, you know, some of them are patient-reported only, and a few of them are clinician-reported. So this is where the clinicians enter all the data into the database. But most of the registries that exist out there are a combination of patient and clinically inputted data. And here you can see the different registries and number of patients that they have enrolled. So this is a rich source of data, but they may not necessarily have collected the data the same way, and standardization kind of becomes a challenge. So how do we approach this? You know, obviously we can't go to these registries and ask them to turn over their data. So the strategy that we thought about was to fund a small scale study that would be enticing enough for other registries to then want to participate in. And so we funded a pilot study using artificial intelligence and machine learning to really dig deeper into the type of data that were collected in the US registry. This is the longest running US registry, the longest patient registry that's been running. The results were published in just very recently, so that we're able to look at medication use and looking for other factors that might contribute to loss of mobility. In parallel, there was a study we were aware of that had looked at the UK registry to see if the manifestation of the disease, you know, as I mentioned, it usually starts in the face, but there are cases of where there's facial sparing, and so patients will develop other weaknesses that spare the face. And so using the UK registry, academic group had really dug into it, and they were interested in seeing if they could validate their finding in using by exploring other registries, international registries. So we funded some additional studies. The first group here, this is a paper I think that's in preparation, and they've integrated what their findings into the UK and the US. So this is the first time that we know of that the UK and the US registries have been analyzed together. And now there's also a proposal that we've received where we are aware of that the French registry and the UK registry and the US registry are looking to maybe combine their data and do a similar type of analysis using AI and machine learning. And just very recently, there was a publication out of China that basically looked at that AI paper, and they tried to apply the same methods to their registry data, and they published the results that were, and there was a really nice response. So I think the data are in line with the findings. So this is one way we think it could help different groups and different curators of registries work together, is to provide them with an interesting approach that they all want to participate in. And hopefully that will pull in additional groups to do the same thing and to partner in these studies. There's also, as I mentioned, the Clinical Research Network is running a lot of longitudinal observational studies. One of the studies that's funded by NIH is called Resolve, and here is a link to the clinicaltrials.gov. For the study, it was designed to enroll 160 patients for a 2-year longitudinal study where they would assess a lot of the clinical outcome measures at regular intervals. The study, I think now is larger. There's a European group that there's European partners. So I think there's about over 220 patients in this. The study has almost completed, and then there is a part 2 to the study where a pharmaceutical company has added additional sites in Europe to add more patients to this group. So that's going to be coming up soon. As new sites were being added to the Clinical Trial Research Network, a new observational study was developed called MOVE and MOVE Plus. And the goal here, this is just kicking off now, the goal is to have 450 patients enrolled over 3 years. And the goal here is very similar to Resolve, to continue collecting data on a series of outcome measures. And here we also anticipate that some of the patients in Resolve are probably going to roll over into this study. So it's very likely that for a cohort, we may have 5 years of data that will be collected. And MOVE Plus is an add-on that was now has expanded to several different pharma groups and different groups that are funding the addition of whole body MRI and biopsies to the MOVE observational study. So this is going to generate a lot of very interesting data. And of course, there's a whole bunch of center-specific studies that have gone on, but one of them that is of great interest is a longitudinal imaging study that was carried out at the Kennedy Krieger Institute, and this is the name of the study on clinicaltrials.gov, where they took whole body MRI imaging for over 2 years with 75 patients. And this is probably one of the largest collections of whole body imaging data that's been collected in FSHD. So how can we get all of these data analyzed? Well, I'm just going to focus on Resolve's data. We have a very good working relationship with the people who manage these studies and the CTRN. And what we did is we just launched a pilot study using a company as a CRO that uses artificial intelligence. And in this pilot study, what we're really trying to get at is can all the different types of data that are being collected, so this is clinical data, patient-reported outcomes, and a host of different outcome measures, can they be analyzed in an integrated fashion so we can possibly identify patients that might progress faster than others? And this is going to be very important as a way of possibly stratifying patients into trials, because as I mentioned, the disease is extremely heterogeneous, and being able to recruit patients with a similar trajectory into a trial will become very important in trying to reduce noise. So this study is currently ongoing. It's just a pilot study, but we can imagine adding to this all of the participants. There's been DNA collected. So at one point or another, it would be great to have the resources to do whole genome sequencing from all these participants. And even though there's probably, the numbers are quite low, the type of data that are collected is incredibly detailed. And it's typically, I think, in the genotype phenotype analyses, the outcome measure that is being assessed is usually age of onset or age at death, something but there's a lot of granularity here in the clinical data that are being collected. And as I mentioned, there's subsequent studies. There's extension of the Resolve with 80 additional patients that could be used. And there's also the 450 patients that are going to be recruited in the MOVE study. That's the follow-up to this Resolve. So this is kind of, we're just setting the stage for possibly doing more extensive analysis. And finally, as I mentioned, there's been several clinical trials in the space. And typically so far, all of the companies that have run clinical trials, they have abandoned their programs. They maybe did not meet the efficacy that they wanted, they were looking to get. So, however, they collected a lot of very detailed and critical, really good clinical data, especially in the placebo arm. And some of these studies were 6 months or longer. So those data would be very useful if we could get them and analyze them. And this is around the time when I became aware of the Critical Path Institute, RDCA, that program. And so we reached out to the companies that had run trials, and here are some of the studies that we were aware of. So Wyeth had run a study, the University of Rochester ran a study for albuterol. There were companies in San Diego, aTyr, that has abandoned. This was more of an inflammatory approach to FSHD. Acceleron completed a phase 2 study, and Fulcrum is just launching their phase 3 study. So they've run several phase 2, both open label and blind placebo-controlled blinded studies. So there's a lot of wealth of data that's been collected here. And so we set up a partnership with the Critical Path Institute, and we're trying to facilitate the integration of these data. And I'm really happy to say that we've contacted all of these groups. All of them are very open to sharing data, and now we're in the process of trying to finalize these agreements. So I believe and Alex can correct me if I'm wrong, but the albuterol data have been taken in. I think we are, as you might know, Acceleron was recently purchased by Merck. Acceleron had okayed the sharing of the data, and I think we need to go through one more round to make sure that Merck is okay with it. We're having discussions with Pfizer now, who acquired Wyeth, and they're very willing to share data as well. And we've started discussions with aTyr and with Fulcrum. And Fulcrum, as I mentioned, they're just starting their phase 3 trial. They are very aware about how important data sharing is, and they are very receptive to sharing their data. They're just trying to figure out when the right time is to do that. So we're looking forward to the participation and their generosity in sharing their data. So just to wrap up, I think the main points I hope to get across is FSHD is a debilitating muscular dystrophy. It's highly heterogeneous and has variable progression. And like many of the rare diseases, it's very important that a better understanding of natural history is going to be really critical for patient stratification, for understanding of the disease itself, and for developing outcome measures that really reflect the disease itself. And there are lots of different sources of natural history and data that exists. And it's really critical to tap into all of them using various approaches to make sure that the data are pulled together and have the maximum benefit in trying to advance clinical development. So with that, thank you very much, and I'm happy to take any questions.
Yeah, thanks a lot, Jamshid, for a terrific presentation. Just to remind our guests here, if you have questions, please type them in the Q&A. We have several panelists today with us. So you've seen her since the beginning of the presentation, but again, Jun Kinoshita, thank you for joining. Jun is Senior Director of Research and Education for FSHD. June graduated from Harvard, started her career as a science journalist with writing in famous publications like the New York Times, Science, and the Scientific American. June has worked in several foundations for multiple neurodegenerative disorder, including multiple sclerosis and ALS and others before joining the FSHD Society. She's been working there since 2012. She also co-founded the Alzheimer's Research Forum, a famous web community for scientists that work in neurodegenerative disorders. So thanks for joining us today, June. We also have Alisa Fink from NORD, our collaborators, joining us today. So Lisa is Director of Research Programs at the National Organization for Rare Disorders. Lisa came to NORD with background in toxicology, but also epidemiology. She has published in rare disease in numerous journals. At NORD, she is Director of Research Programs. She oversees strategic development and implementation of scientific activities to ensure that NORD drives innovation to support rare disease communities. She's also very active with RDC ADAP and help us drive a project as part of the rare disease program at CPAF, which is a partnership with NORD. So thanks for joining us today, Elisa. We also have Megan. Hello, Megan. Megan is Quantitative Scientific Director for RDC ADAP, and she's also with us today. So Jamshin, maybe just to kickstart the conversation, and then we'll jump to some of our attendees' questions. You talk extensively about this meeting in 2019. I know you also had a more recent meeting this summer. I was just wondering, how has it evolved in the last couple of years? What are the key outcomes of the meeting you had recently with industry, and also maybe if you could comment on the industry perspective on what's needed for drug development for FSHD. But maybe let's just start with kind of the...
I'd be happy to. Yes, obviously COVID kind of put a little damper on our ability to host in-person meetings and have, our goal really was to have these industry collaborative meetings take place on an annual basis. But really, I think it's important to have these face-to-face, and we were lucky enough in June to be able to pull one together. We had representation from about 16 different companies join. There was also, I think, representation through virtual participation from at the FDA. And we basically went through all of the gaps that were initially mentioned. So from the biomarkers, and the CTRN outcome measures, and we discussed all of that with KOLs. I think what was really different was in the first meeting, we were listening to the different companies tell us what the perceived gaps were, and the gaps are still there. In this meeting, we actually gave a lot of the presentations on activities we were doing in trying to address the gaps. And some of these, including one of them, for instance, for genetic testing, we created a platform where about 5 or 6 different companies have provided funding, and we are making genetic testing available directly to patients. As I mentioned, the genetics are quite complicated, and you cannot use next generation sequencing to identify patients. And one of the obstacles in recruiting patients into clinical trials is they need to have a genetic confirmation of the disease. And that's typically not possible because either insurance doesn't want to pay for it, or many neurologists don't know how to order the test. It's quite complicated to order it, and there's only 1 or 2 sites that provide genetic testing for FSHD. So we created this direct to consumer, direct to patient testing facility where they get genetic counseling as to whether the test is appropriate for them or not, because it's going to go on their medical record. And then it's fully sponsored, so they will get the phlebotomy is done at home because many of our patients have a difficult time with mobility, and so you have in-home phlebotomy, and then the testing gets done and reported back to them through a genetic counselor. So we're just running this as a pilot study, but I think this is something that industry sees as a benefit, and they will probably continue to fund it. And then we discussed a lot of the biomarkers is still an incredibly important measure. that is needed, and there are different ways and different approaches that academics and industry has taken. And so there was lively discussions on, let's say, how to, what kind of imaging should be done. And from that, we're hoping to develop a project where maybe the same data sets can be analyzed using different methods and compared to each other. And ideally, all of the different methods will point will give the same result, and then it becomes it'll be irrelevant. Anybody can use whatever method they want as long because it will give consistent results. But those type of studies haven't been done yet, and these are the type of studies that industry will probably support. And so we can help offset the cost, and it will de-risk it for everyone. Excuse me.
Great. It seems like you're really fostering a very collaborative environment with industry and aligning the community, which I think is always critically needed.
It has been absolutely fantastic. Not only have the industry partners been very collaborative, but a lot of the academic groups that are in the space have also been very willing to share and collaborate. And nothing compares to the willingness of the patient community. They are really at the core of everything that we do. And they have, whenever we send out a survey, we get unbelievable responses. They're a very motivated group, and they really provide a lot of input and participation in all of these studies. So that's been really phenomenal.
We have a lot of audience questions, so I think we can start addressing these. Megan, do you want to maybe start? And
Sure, we'll be glad to. So the first question that came in is the USMD StarNet Data Hub considered a registry for FSHD patients?
Sure. June, do you want to take that?
Okay, yeah, I'm happy to field that. So I confess I don't have a deep knowledge of what's in the MD StarNet database, but my understanding is it's data from I think Medicare, possibly Medicaid patients in the state by state basis of the states that are participating in the study, and so it's not structured in the same way that like registry data is. For example, I don't believe that that these sources collect data using the core data sets that have been agreed on for international registries. So they're very valuable in other ways. They're a kind of real world data. There's a lot of claims type data in there. It complements what's in the registries, but I don't think that it would be considered a registry per se.
thanks, June. And I have a question from Teresa Brandt, discussing the study by Katz and al. About the predictors of functional outcomes using AI. So the question is, what do you perceive to be the remaining opportunities and limitation to using AI in the setting of FSHD in the RESOLVE study? There's a follow-up question about the involvement of FDA, and did FDA weigh in on the plans, or were you able to get any feedback from FDA? So maybe if you can start answering, Jamshin and Megan also, if you want to comment on, the power of AI, the power of disease modeling applied to understanding disease progression, especially in the context of a disease like FSHD with different genotypes, a lot of heterogeneity between patients, and also between different genotypes, but also within a genotype between patients.
Sure, I'm happy to take a stab at it. As I mentioned, I think when we first started this project, we were using AI as an approach to get other registries interested. I worked for quite a number of years on Huntington's disease, and Huntington's disease also had different patient registries scattered throughout the world, and it was quite challenging to get all of the registries to provide data for integrated analysis. So one of the advice I had received from my former colleagues working on Huntington's disease was, if we create a project that's interesting enough, other groups will want to participate in it. And the power of it was the US registry is definitely the number of participants. I think there's 800 or so patients in the registry, maybe 1,000, but about 600 of them have genetic confirmation of the disease. So I think the study used just a genetically confirmed patient. It's quite underpowered. I think most AI studies that are looking at genotype, phenotype they require thousands of patients. But this is just the start, and other groups have responded very positively. So this is something that we're hoping is going to snowball, and as more groups get interested, it will become possible to do these type of broader analyses. And Resolve is the same way. It's quite limited with 200 patients, but the data that are being collected are quite granular. And as MOVE, I think in the next iteration of the study, family pedigrees are going to be collected, I believe. I believe that's true. So that's going to add another level to help with the analysis, especially if we try to do genotype, phenotype analyses. But I'm happy, I'm not an AI expert. I don't know anything about the statistics of it. So I rely on KOLs and different groups to guide us in this. And I'd love to hear from other people who might be on the panel and have experience in this area.
I can just speak to it a little bit. So neither am I an AI expert, and I'm not familiar exactly with the Resolve study, but what I can say is with these types of, in general, these types of analyses, it really facilitates, maximizing the data that's available. So if you have genotype, phenotype, you can really use that information to identify, maybe some subpopulations within the patient groups that exist that may respond better to certain types of treatment. So, it's very, powerful tools that can really accelerate the drug development in this space.
Yeah.
So I think we have maybe two questions that are a little bit related to getting access to the data on the platform. So one related to patient consent. So are you running into issues with patient consent for downstream use of clinical trial data, or was that already considered in those consents? And then related to that, what are the biggest challenges in getting the industry to participate and share data? And what are there any incentives?
Yeah, I've been really delighted by the industry response. They all of them have experience in the rare disease space, and they all see the benefits of data sharing, especially a placebo arm of a study that they carried out. So even with some of the people that we approached, the team that ran the clinical study in FSHD are no longer there, and there's a brand new team at the company. They were very open and willing to do the data sharing. I think where we're getting where things are slowing down is when it gets to legal and getting the legal agreements. And we're just touched on it in the previous question, the consent. I think a lot of the legal groups are looking at the original consents to see if data sharing would be possible or not. I think so far, the challenge has been trying to find all of those forms, because a lot of the studies were carried out maybe 5-6 years ago, and it's very difficult to find the informed consents for each site, and getting that paperwork ready for the legal to review has been challenging. But Alex, I think you've had some also experience with different groups. What has been your experience?
Yeah, I mean, the consent is always, for us within the RDC Lab platform in that context about sharing data to us, like the real block for sharing data, which can prevent, an industry, for example, to share a data set, is determination on whether the consent allows for it. So especially for legacy data, it is sometimes something that comes up where industry tells us we can't because of consent. When you talk about legacy and sometimes old trials, there's always a pending question for us, I think, is it really an issue or not? But it is one of the blocks that we're facing within the context of the program. On how to motivate industry to share data, maybe Elisa, if you want to comment on that, because I know Nord is also actively engaging with a lot of industry and communicating about the program. So maybe do you want to give your perspective on how to motivate industry to share data?
Sure. I haven't been as directly involved in some of the industry conversations, but I think, as time has gone on, people really are understanding and appreciating the value of this data. I think, in this data world that we live, people understand the power and the value of data. And, increasingly people are sort of trying to find ways to do it. And I think from NORD's perspective, as we have had the opportunity to really empower patient advocacy groups to collect data and to leverage this data, and slowly they're having the opportunity to work with industry and collaboratively sort of think about, what's possible with this data and what can we do collaboratively, but recognizing that there are concerns around privacy and consent, and those are some things that we really need to think about, and we really need to think about potentially a priori as we start developing and designing studies.
So a little bit tangential to that, Eliza, a question that came in. As both a patient as well as a research scientist, is there a way to get involved in performing research to better understand the disease cause development as well as therapeutics development?
Sure, great question, thank you. And so just to give you guys just a little bit of perspective, NORD is a membership organization. We currently have about 350 patient advocacy groups who are part of NORD. And so as a group that's really thinking about how do we support this entire rare disease community, we think a lot about building tools and resources and opportunities to really help people engage in this research process. But we also recognize that thinking about research and research readiness is really multifactorial, and there's so many pieces that are involved in this, and that for each group and for each disease, different things are needed. And so just to sort of give you some examples of some of the tools that we have been thinking about that may be relevant in your world. So the first one is we've realized the drug development process is complicated, and there's a number of steps, and unless you're really enmeshed in it, it's not there's a lot involved. And so one of the things we've done is, as part of our learning management system, is to create a research ready series of information and education, so people, to really empower people to really understand, like, what is this process? Where are the places that I can get involved? What are the things we need to know to really level set so that everyone's coming into this with equal knowledge? And then specifically, NORD has developed what we refer to as our I Am Rare registry program, which is a combination, which is in all cases led by individuals and patient advocacy groups who really sponsor a registry or a natural history study up to them on the platform. And really, it provides both a tool for data collection, but even more so, a program and a community of people who are all thinking about collecting this data, with us providing support around the regulatory, and the steps involved in doing this. But one of the things we really want them thinking about as they design these studies is, what are the things that are important to the patient community? What are the things about disease progression or the lived experience of that disease that are necessary to collect because they are important and impactful to patients? And then thinking even more about sort of like thinking about things like clinical outcome assessments. What are the things that clinical trials need to measure? Because that's how we show success. What are those things that we need to think about? And so, like, all of these things sort of coming together, I think, are all opportunities for individuals and researchers to really think about engaging in the research at various levels in various ways. And I'm happy to talk more if folks want more detail, but I realize we're running out of time. So thank you.
I can just briefly jump in. We have a lot of people in the FSHD community who are scientists or want to get into careers in science and medicine. So I would say, if you have that interest, reach out to me or Jamshid. We are constantly matching people who want to get involved with the labs. We want to know what your background, your interest is, and then we can think about what labs might be out there that might welcome, having you as a member.
Thanks, June. Well, thanks, everyone. I think just to wrap up, again, thank you for this presentation and terrific Q&A. We've been discussing a lot throughout the last year, Jamshid and Jude. Just maybe a last thoughts on, so we're starting to get trial data into RDC ADAPT. There's a lot of registries effort. What should we be doing to take it to the next step in aggregating more data for RDC ADAPT and maybe starting some work? Do you have any like final thoughts or comments?
Yes, I think as we get more data in this, definitely the first thing is to gather the data. And then actually, I saw there was a question we didn't get a chance to address by Tina Duong on making sure that the type of clinical data that's being collected actually can be compared apples to apples, because quite often they may be collecting data differently. And I think this is, we're going to be relying on the expertise at the RDC ADAPT for the standardization, but also from KOLs, and getting the KOLs and the clinicians that are collecting these data and developing the methods involved in the analysis is going to be really critical. So we're hoping to get people like Tina involved as well through a project. So that would be the near-term goal. Yes.
Yeah, and I have to say the informed consent issue is one of the most vexing to me, because almost any patient you talk to will be shocked that their data is not openly available, especially anonymized data, which is what we're interested in. And you know, so first of all, you know, if you're writing informed consents today, make sure they have, you know, broader applications. They used to be written more narrowly, but also often I feel like these companies, that just legal people are just kind of, you know, doing, I don't know, what polite way to say it.
Well,
but if there's a creative solution, maybe, you know, maybe ask all our patients to reconsent for all the studies you've ever, you know, or something, I don't know, because you can go down a rabbit hole for years just haggling with these legal departments that are dealing with something that happened multi-generations and multiple companies ago. And it's very frustrating.
Thanks for that perspective, June. And I do share that frustration. We're currently working to try to bring some solutions about the consent issue. But the idea of prospectively engaging with your community, whether FSHD or any rare disease communities, and talk about the consent and the need to build in consent whenever there is a data collection effort is really critical. So thanks again, everybody. I just want to announce that RDC ADAPT, the Rare Disease Cures Accelerator, will be holding its annual workshop September 13th and 14th in person in DC. We will also live stream the first day of the event, but it would be amazing to have you join the meeting, and people that can make the trip. We welcome you to attend our great meeting that has been in preparation for the last several weeks. So thanks again for attending this webinar, and hopefully I'll see some of our live attendees at our workshop. Thanks, everyone.
Thank you
very much.
Thanks, everyone.